My name is Bingwen, and today I'm presenting about tissue mineral density distribution in human mandibular premolar region. So the alveolar bone surrounding teeth is actively remodeled as a result of bearing repeated high-impact dynamic masticatory forces as we chew and eat every day. So the constant bone turnover results in different levels of mineralization of bone tissues, and this active bone remodeling causes more mineralized pre-existing bone tissues to be replaced by the less mineralized newly formed bone tissues. This process makes the alveolar bone has higher heterogeneity of tissue mineral density than bones at other sites. And the hypothesis is that the dynamic masticatory forces have an influence on the local tissue mineral density. Our specific aim for this study is to examine how large of an area of alveolar bone is influenced by active bone remodeling. Let's talk about the method that we use for this research. So the human mandibles were obtained from 5 cadavers of human mandible ranging from 43 years old to 80 years old, and none of the samples had dental restorations such as composites or amalgam, and the premolar region of each specimen was sectioned with a low-speed bandsaw. The picture you see on the right was taken right after I cut the premolar out of the whole mandible. Each specimen was scanned using the microcomputed tomography with 27 micrometer voxel size. Then we used the hydroxyapatite phantoms with known density values to calibrate each CT attenuation value of the voxels and then convert them to mineral densities. So after the sample or scan, we use the ImageJ and model prep software to separate the bone voxels from the non-bone voxels of the tooth and the soft tissues. And then the teeth were digitally removed, leaving the alveolar bone isolated. The sequential three-dimensional layers of the alveolar bone were isolated up to 3.2 millimeters from the PDL, and each layer has a thickness of 1 voxel. And we obtain a histogram of the tissue mineral density from each layer to evaluate its mean, standard deviation, 5th percentile low, and 5th percentile high. And we use a paired t-test to compare values of the tissue mineral density parameters and variation between individual layers. Let's talk about the results that we got. So the mean values of the tissue mineral density rapidly increased from 54 micrometers to 108 micrometers from the PDL and were significantly lower than further layers. Other than that, all other layers, all other tissue mineral density parameters were not significantly different between layers. So the first question that we have for the discussion is how masticatory forces influence local tissue mineral density. So the findings in this study indicate that masticatory forces likely influence local tissue mineral density distribution at the alveolar bone region. And this agrees with the findings of previous studies that alveolar portions tend to have a reduced mineralization density. And these findings may have clinical applications in orthodontic or periodontal treatments as the alveolar bone provides significant support to teeth and can impact tooth movement. Some limitations of this study. The biggest limitation is that the number of specimens were not sufficient for conclusive evaluation of bone mineral density. And it has been found in previous studies that each area in the dentition demonstrate unique regional variation, which means that the results of this study might not be applicable to other areas in the mouth, such as the molar or the incisor regions. So to address these limitations, I think future studies should involve a larger number of bone specimens, and further investigations are needed to determine the tissue mineral density in different regions of the mouth. In conclusion, the high-resolution micro-CT-based TMD analysis allow for mapping the detailed mineral distribution of alveolar bone next to tooth, and the current result could visualize the mechanobiologic response of bone to applied force. And I would like to thank my mentors, Dr. Dogu Kim and Jia Liu, and my partner Amir Hasuna for making this research possible. And here's some of the references that I use. Thank you so much for listening.